Hi, I'm Anna, a member of the UCL iGEM 2020 team. So our team is made up of 8 undergraduate students and 3 supervisors. All of us are contributing to our webinar sessions over the summer. Just to quickly go over review our activities. So we had our first session 2 weeks ago talking about molecular bio, natural sciences, and immunology. And a week ago, we went over biochemistry, and today we're presenting biochemical engineering. And for previous sessions, they are available on our YouTube channel. So if you go and search up UCL iGEM 2020, you should be able to see all the casts we had for previous sessions in case you missed any. So now let's meet our speakers that presented in previous sessions.
I'll start with introductions. Hi guys, my name is Pedro, and I'm a second year molecular biology student. And yeah, I'm really enjoying iGEM.
Hi guys, my name's Juliet. I'm a third year natural sciences student. I hope you enjoyed the first talk, which was now 2 weeks ago. And I hope you enjoy this one as well.
Thank you, Pedro and Juliet. It was a great presentation that we had 2 weeks ago, and we don't have Professor Kent and Stefan today joining, but they were also introducing molecular biology and immunology 2 weeks ago, so please check out our YouTube video to see what they've talked about. And now moving on to the speakers for session 2. Please introduce yourselves.
Hello everyone, my name is Daniel. I'm a third year biochemistry student. I joined iGEM because I liked its multidisciplinary aspect. I was the speaker for the previous webinar, and I hope you enjoyed, and you can find it on YouTube.
All right, thanks, Danny. So as you just heard from him, we were speakers in the previous webinar. That's up on YouTube. Hope you guys enjoyed it, and now we're gonna hear from the engineers. So I'm Ollie. Thanks for coming.
Thank you, Ollie and Daniel. And Alex, our PhD, was also part of the webinar 2 speaker, but he's not here today. So now moving on to today's session. Hi everyone, I'm Anna, a first year biochemical engineering student at UCL, and I joined iGEM because I wanted to learn how programming could facilitate SynBio research. And besides bio, I'm also passionate about coding and robotics. So please introduce yourself, Laide.
Hi everyone, I'm Laide. I'm a second year biochemical engineering student, and I'm interested in computational modeling and how we can use that to study biological systems. And I hope you enjoy the webinar today.
Hello everyone, I'm Lee, a second year student at UCL majoring in bioprocessing of new medicines with business and management. I'm interested in how to commercialize biological research, improve biopharmaceutical manufacturing and marketing, and any other bio-related stuff. Hope you enjoy today's webinar.
Hello, my name is Stefanie. I'm a lecturer in synthetic biology at the Department of Biochemical Engineering, and I also do research in synthetic biology, and I'm the team leader of this year's iGEM team.
Thank you, Steffi. And today we have one more speaker. So that's Rana from our department, who's gonna introduce our major today. So now let's move on to, I'm gonna quickly go through what we are talking about in these webinar sessions. So basically we're gonna introduce the majors that we are studying. That includes biochemistry, molecular bio, biotech, biochem eng, immunology, and natural sciences subjects such as medical physics and neuroscience. And basically in our 3 webinar sessions, we went over, or we are gonna talk about what these majors include, like what core modules we're taking in them and what their applications are, as well as their potential career pathways. So hope you guys all enjoy that. And now let's talk about biochemical engineering, and I will hand it over to Rana to speak about that.
Hi everyone. So my name is Rana Khalifi. I'm a senior teaching fellow in cell and gene therapy or regenerative medicine, biochemical engineer. Originally, I'm not an engineer, but I'm happy that I did my PhD in biochemical engineer, as I will tell you why in the next slide. So the first thought that you think about, what's biochemical engineer? I usually get a lot of questions related, what is biochemical engineer? Is it the same as chemical engineer, or is it related to biomedical engineer? Well, it is different. Sometimes it can cross with chemical engineer in some of the terms and biotechnology, but basically biochemical engineer is an interdisciplinary major, which is great because if you like, for example, physics, chemistry, but you're interested in medicine or biofuels, you want to incorporate business with the engineer, with like the normal treatment for stem cells, let's say, but so let's say you're interested in regenerative medicine, but it costs a lot. So how can you, using your knowledge as biochemical engineer, reduce the cost of the therapies? So basically, we are interdisciplinary course, and as I will show you in the next slide, we have a lot of research going on in our department. So briefly, I will try to explain to you. So if you're interested in doing undergraduate at biochemical engineer, we have the normal biochemical engineer, BS in biochemical engineer, which is BEng. Or if you want to do 1 year extra, which is you will have a master's in engineer, MEng. And we started recently, a few years ago, a new courses called bioprocessing. And we have 2 streams in bioprocessing. So we have the science and engineering stream, and then we have the business and management. So these 3 streams, although they are in the same department, they differ depending on what you want to do. For example, the BEng will focus more on the fundamental explanation of the engineer related how you can design a bioreactor, the thermodynamics in it, and then how you incorporate your knowledge to reduce the cost, for example. And then what's good about it is that within the Faculty of Science, you have you can take a minor, so you can select minor from our department, or let's say you do biochemical engineer, but you do a minor in let's say environmental science. So you can choose whatever you want. The good thing about the MEng is on your 4th year, you have so many options, like going study abroad, or the most important one is to do 1 year in industry. Which you will gain a lot of experience, and maybe you will come back for postgraduate. The bioprocessing, nowadays we had a meeting with industrial people, and they said we're requiring more or less 300 people per year, which with the bioprocessing knowledge. Okay, but I'm not gonna go more into details. If you would like to have more to know more about it, and then the requirement, the GCSEA level requirement, please go to our website, Biochemical Engineer. Moving on, so this is basically what I told you about. You have so many options. You can do a year in industry, so you will be working while doing your MEng. You will come back to us for a few days for short courses, or you can do study abroad, or you can do a collaboration with a chemical engineer and business and management. Now, if you choose to study biochemical engineer, as I said, because you're going to be focusing more on the thermodynamics, fundamental knowledge of engineer, you will have many courses, and you will have a variety of the courses. So you will learn about, let's say, mathematical model using, let's say, MATLAB. You will know about how to design synthetic biology, for example, with Steffi. You will learn about molecular biology and biochemistry, although also with Steffi, heat mass transfer, thermodynamics, and now the hot topic, which is the vaccine bioprocessing. So as you see, we cover most of the topic and hot topic nowadays. Now, if you choose to select the bioprocessing, Next slide, please. So if you selected the bioprocessing, more or less, you will have similar courses like the vaccine, for example, and then the regenerative medicine minors that I teach. But you will have, if you're more into the business and management, you will have courses with the business and management department to tell you about the financial accounting, and then you will have the most important one is the design project, in which we will give you kind of a scenario in which you will go and investigate and see how you can make the bioprocessing in a better way using the finance. So you're gonna have like cost analysis, and then at the same time incorporating your knowledge and engineer in order to modify the process. Now, although we're a small department and we just started like more than 20 years ago, we have a lot of variety in research. So we have one group who's focusing, let's say, on the vaccine. We have a Vax Hub, which we collaborate with a lot of universities, other universities like Oxford and even AstraZeneca, and that they're leading now the vaccine in the UK. We have something called regenerative medicine, or what we call now cell and gene therapy, in which we focus, let's say, on tissue engineer. What is the newest biomaterials that you can use in tissue engineer, like the skin, like the bone, for example. For me, I teach this, and I teach also stem cells. So how can you, for example, take a cell, make it stem cells, and then differentiate it into, let's say, retinal cells and give it back to the patient? Or nowadays with the trending CAR-T, which is the new therapy for cancer. So how can we optimize this process to make it affordable? So with biochemical engineer, what you need to think about is, with the faculty of engineering, we say how to change the world or how you can help the world. So it's nice to have some research and everything, but how can you make it affordable to all the people and make it available for all of the people? So this is one research group. Another research group is focusing, let's say, on biofuels. So how can you grow algae, for example, to produce bioethanol or to reduce the pollution? We have a group working on plastic, and of course we have the synthetic biology group. Now, it's super nice to see when you graduate from biochemical engineer and you go to any conference or you go to a meeting, you see all of your colleagues, because basically most of our undergraduate, postgraduate, even PhD students, when they will finish, we have a target industry or pharma where they go and work. So you have a lot of variety. So for example, some of them, they will just go and work with GSK, like the big pharma, or some of them, they will go to consultancy or business plan. So you will have a lot of variety as career once you finish, because you've learned so much. And the way we teach is not only lecture-based, we run scenarios, we run case studies, so you will be familiar with most of the cases happening in the industry. For example, during year one, we'll take you to a ice cream manufacturing in which you need to optimize their bioprocessing. So more or less, you will have contact with the industry, and then you can see if you fit there or not. Or during the second year, you will have like kind of a pilot plan in which you will go to the lab and experience lab-based experiments, so you will know what you want. So most of our graduates will go to the companies that we collaborate with, but as I told you, you can go to consultancy and other biotech companies. I think that's all from me. Thank you.
Thank you, Rana, and thanks for introducing our majors. It was a great presentation from you. So now moving on, me and my teammates and also our supervisor, Stefanie, we're gonna talk you through our journeys of how we entered the field of biochemical engineering. So I'm gonna start with my journey. As I said before, I'm a first-year undergrad studying biochemical engineering at UCL. And my journey actually started pretty young, like when you see this little picture of me, so basically when I was in high school, I was super interested in a variety of STEM subjects, like coding, and then in maths, I was really into calculus and geometry. And in sciences, I particularly enjoyed studying molecular bio and chemistry, but at the same time, I also enjoyed doing engineering. So I was really looking into a major that could integrate all of my subject interests. And fortunately, I found out that biochemical engineering, it's a perfect degree to integrate all these interests. And so what do I like the most about this degree? Well, it's actually the interdisciplinary aspect of it. And what I mean by that is, for example, if we look at this, it's called the piping and instrumentation diagram, PNID diagram, that I drew in one of my first year modules. And this integrates both bio and engineering. So for example, if you want to culture cells in this vessel you see in the middle here, it's called the bioreactor, then what are the parameters you have to consider in order to have a suitable growing condition for your cells, right? Then you have to consider parameters like the pH, your oxygen level, and also temperature. Those are just examples. And the engineering aspect is, what do you do with those parameters? How do you measure them, right? So then, for example, for oxygen, you would have this probe to measure dissolved oxygen. And then you would have a computer to control this parameter. So it's going to send signals to a valve saying, okay, maybe we need more oxygen. So yeah, this diagram, I think it perfectly shows how the interdisciplinary aspect it's included in my major. And besides that, I also like the wide applications and future career choices. As Rana said, there are so many pathways that you can take after graduating with this degree. Something that I haven't seen so far, it's that I don't really know the connections between certain modules that I had in first year. For example, I haven't really seen how thermodynamics is related to, for example, bioprocessing or fluid flow that I took. But I've been talking with second year and third year students, and they were saying I should be seeing more connections between my modules in later years. Just a little bit about my research interest so far. So I'm particularly into this field called protein topology. Basically, it's an interdisciplinary study of math, computer science, and biology. And it studies the property of a protein that remains unchanged without breaking up the structure. So what it means by that is, in topology, you don't consider the size of something, you only consider the shape of it if you don't break it up. And that is actually based on an idea we all learned in high school biology, is that protein shape is related to protein function. So by identifying the mathematical knot type of a protein and study how that is related to its protein function, it's going to be really contributing a lot to molecular bio research and protein studies. So how do you really identify the mathematical knot type of a protein? Well, here, please look at this globe showing on the bottom right here. Basically, it's within this globe, it's going to be a protein structure embedded in it. And as long as you look at the structure from any point that's in the same region of the color, then you should be seeing the protein structure having the same mathematical knot type. What I mean by that is, for example, if you look from any projection point from the green area, then you would see the protein structure showing, as we see in the key here, it's saying a 3.1 knot, so a trefoil knot. And what it looks like, if you go to this knot type table at the right here, the trefoil knot is it's this one, the 3.1. So how do we go from protein backbone structure to its mathematical knot type? Well, you are gonna first look at the backbone of a protein, and you are gonna get the X, Y, and Z coordinates of your carbon alpha atoms from the protein database bank. You're gonna import all that data into topology tools. For example, the one that I used was called Knotplot, developed by Rob G. Sharan. And so now moving on, just gonna show you guys a little program that I made in 2018, and this shows exactly how a protein backbone structure can be simplified to its mathematical knot type. So as you see here on the right side, I have loaded some data from PDB, and I coded them so the protein structure is loaded in this topology tool. And when I click on go, And then bead, it's going to simplify the structure to its mathematical knot type. As we see here, again, this is called the 3.1, the trefoil knot. And after I made this program in 2018, another paper was published introducing another approach called knottle ID, which tells you how other ways that protein chains can be simplified to study its mathematical knot type. And if you are into protein topology, I would highly recommend you to check this out. And this is also my current interest of study, so to figure out how this knottle ID works. And so what do I want to do in my future years? Well, starting second year, as Rana said, we engineering students could pick a minor to study along with our major. And I'm going to do intelligent systems, so probably going to be like this kitty, just coding in front of a computer all day long doing artificial intelligence. And after undergrad, I will look into a PhD in protein topology. However, I have no idea where I'm going to do that, so do need a lot more research into where I want to do this. And afterwards, my plan is to go into academia and to pursue this idea that computer science, it's a bridge between biology and maths. What I mean is, for example, as you see in protein topology, you have to use computer science in order to connect the information in biology into along with the information from maths. So basically, I would be really interested into studying the interdisciplinary aspect of protein topology. And finally, just a little piece of advice from me, I would say to keeping heart not just in mind, that if you are passionate about many different STEM subjects like me, then definitely don't limit yourself by only going into one direction and forgetting all the others, because you will eventually see how your academic interests are all linked together, and you will see how the interdisciplinary study of them contribute to tackling real life challenges. So thank you all. I hope that was helpful. And now I'm going to pass it on to my teammate Laide to talk about her journey.
Great, thanks Anna for that. That was really interesting. So as I introduced myself earlier, I'm Laide and I'm a second year undergraduate student studying biochemical engineering. So my journey into STEM began with my interest in maths, biology, and chemistry in high school. And when I was deciding on what I wanted to study at university, I wanted to study a degree that combined all these subjects. When making my decision, I came to a point where I had to make a decision between science and engineering. And how I saw that was that science involves observing and understanding the natural world. Meanwhile, engineering involves applying the knowledge that we've gained from science to solve problems. And in high school, I was really interested in problem solving, which is the reason why I chose to study engineering, and I'm now studying biochemical engineering. So what do I like and dislike about my course? I like the multidisciplinary nature of the course, and Rana and Anna spoke really well about this earlier on. The degree does involve the study of maths, biochemistry, as well as physical concepts like thermodynamics and heat and mass transfer. We also study business-related topics such as business law and also cost analysis for commercializing products and designing bioprocesses. I also like that there are a lot of real-life applications and opportunities for problem solving. So as Rana talked about earlier, we have scenarios where we're presented with problems where we have to design processes and come up with a solution to these problems. What I don't like as much about the course is that there isn't a large emphasis on learning the scientific details. I personally am a detail-oriented person, and I do like to know a lot about the scientific details, but the focus in the degree is more on engineering design and calculations. So my research area of interest is in computational modeling and how we can study biological systems and how their environment affects their characteristics and behavior. So in biochemical engineering, computational modeling can be done on the level of the cells and also the environment that they grow in the bioreactor. So in biochemical engineering, we basically view cells as manufacturing factories, and we want them to be as efficient as possible and produce as much product as possible. So we can have a systems biology approach where we use new technologies to develop computational models to understand how biological systems work. And these insights that we get from the models can be used to design improved strains that will be more efficient and produce more products. We can also model the environment in which the cells grow in. So as Anna mentioned before, we want to have a constant environment where the cells can grow. They need to have sufficient access to nutrients and also oxygen and have the suitable temperature for growth. So an important thing, a problem that we do look at in biochemical engineering is how we can optimize the mixing of the bioreactor where the cells are growing so that they can have the optimal conditions for growth. And as we work at very large scales of thousands of liters, mixing becomes more non-trivial, and it becomes a lot more difficult for us to have these homogeneous environments where the cells can have access to all the nutrients and oxygen that they need for growth. So instead of doing experiments at very large scales, we can have models where we model the fluid flow in a bioreactor, and this can inform the process design so we can optimize the process to have sufficient mixing and mass transfer of nutrients and also have an appropriate temperature conditions for the cells to grow. So for my career plan, I'm not 100% sure on what I want to do yet, but I'm interested in the option of doing a PhD or an ND, which is similar to a PhD, but you work together with a company or with the industry. And I'd want to work on a research topic related to computational modeling, but I'm not quite sure which level I'd want to work at yet. I'm also interested in working in the biotech industry, specifically in biopharma with the development of new biopharmaceutical products. But as I said, I'm not quite sure when or which is going to come first or the order in which this is going to happen. So my advice for choosing a course at university is to study something that you find interesting, and remember that there is an overlap between some of these degrees. When I was choosing what I wanted to study at university, I was obsessed with being certain of my choice, but in reality, you can shape and tailor the experiences you get throughout university, and what you study to become may not be what you choose to become in the end. So I'd say that you should choose a degree that you really enjoy and be curious enough to explore beyond the content of your degree. Now I'll give it over to Lee to talk about bioprocessing of new medicines.
So thank you, Rana, Anna, and Laide. So hello everyone, I am Lee, a second-year biochemical engineering student at UCL, majoring in bioprocessing of new medicines with the stream of business and management. Nice to meet you here in session 3. So before I start, let's consider the differences between pharmaceuticals and biopharmaceuticals. Both of them are used to treat specific diseases. For example, aspirin, which you might be familiar with, is a pharmaceutical used for reducing fever and relieve mild to moderate pain. On the other hand, Herceptin, which is a biopharmaceutical, is used for treating breast cancer. So what are the differences between pharmaceuticals and biopharmaceuticals? First of all, their molecular sizes are totally of incomparable scales. As for aspirin and Herceptin, they are composed of mere 21 atoms and more than 25,000 atoms respectively. So you can imagine that the molecular structure of Herceptin would be far more complex than that of aspirin. As you regard aspirin as a bike, then Herceptin would be an aircraft. Moreover, normally for biopharmaceuticals, they are produced by living cells within a bioreactor with multiple extremely complex bioreactions. some of which you may not even to predict what would happen within the bioreactor. However, pharmaceuticals are formed mainly via chemical reactions. This indicates manufacturing process for producing biopharmaceuticals and pharmaceuticals are totally different. And due to reactions are taking place in living cells for biopharmaceuticals, uncertainties are added to the process. Despite that, biopharmaceuticals are more effective than pharmaceuticals in treating diseases, especially severe ones such as cancer and central nervous system disorders. So after clarifying this, I would like to share with you some of my journeys. In addition to being a biochemical engineering student at UCL, I'm also an ACCA student, which stands for Association of Certified Chartered Accountant. So besides what I have learned in uni, I also have some professional skills in accounting, finance, economy, and investment. Finally, before I come to UCL, I have studied Italian for 3 years during my high school. And at that time, I also had a chance to go to Italy as an exchange student. Regarding my course, it is a well-designed interdisciplinary course. As you can see, I study not only biochemical engineering courses, but also business and management courses. However, I would say business and management courses are a bit different from other courses because contents in these courses are usually abstract, which you might even question yourselves. What am I learning? What's the point here? But actually these courses, instead of directly teaching you something, they are influencing you. Sometimes you are using your business acumen to think about other things even without your consciousness. Also, in this course, you would know how to convert your idea into money. However, as you split some of your time learning business and management, you would not have enough time to learn deeply the principles of some specific biological knowledge as what my teammates demonstrated before. So you have to consider the balance. So my research interests are mainly in two aspects: bioprocessing of new medicines and business and management. First, bioprocessing of new medicines, as what you can see here, are called upstream and downstream processing. As what I have said before, biopharmaceuticals are mainly produced in bioreactors. However, it is not as simple as putting cells and substrates in the bioreactor and we are done. No, definitely not like that. We have to conduct experiments in different scales before we can finally produce them. From lab scale, which might only be several milliliters, to pilot scale, which can be about 30 liters, as the one you can see here. So here, the bioreactor is the same as the one in our biochemical engineering lab, and it finally goes to production scale, which can be as huge as 30 liters. For downstream, it is the process in which we extract proteins in the bioreactor produced by cells to form the final products. Since what we are producing is drugs, which would be used directly on human bodies, we have to ensure their purity and select the most appropriate administration methods, either via oral, injection, nasal spray, or other methods. However, in reality, upstream and downstream processes are far more complex than what I said. Another research interest comes from the aspect of business and management. As you can see the diagram here, biopharmaceuticals are getting more and more market share and generating more and more revenues from 2010 to 2024, which I suppose one day it might take the place of pharmaceuticals and mainly dominate the drug market. However, the cost and time of launching a biopharmaceutical product is huge. It is said that it takes 12 years in average to bring a biopharmaceutical from R&D stage to finally on the market. And this process usually requires billions of dollars with a success rate of only 12%. As you can see, for a drug to be qualified to be manufactured, it has to pass multiple stages such as research, preclinical trial. Thus, biopharmaceutical industry, although it can generate huge profits for companies, not many investors are willing to invest due to the high demand for capital but low success rates. Now let's move on to my career plan. Basically, I separate it into different stages. I'd like to choose to work in pharmaceutical consulting or work for large pharmaceutical companies, especially those investing heavily in biopharmaceutical development, such as Pfizer, Novartis, GSK after I graduate from my later master's degree. Then perhaps I would start up my own business, probably in the biosimilar industry based on the experiences I would have gained by then. If you guys want to know further about biosimilars, welcome to discuss more with me, and I might also share some ideas on currently how large pharmaceutical companies overcome capital requirements in biopharmaceutical research and development. So finally comes my advice. From the perspective of a STEM student, I would strongly recommend you to find where your real interests are. While from the perspective of a business and management student, although you should locate your interests in specific fields or industries, it does not mean overlooking other fields with which you are not familiar. Always stay open, proactive, and diverse. More importantly, practically, I would suggest you gain as many social experiences as possible through whether volunteering or internship at your early stage if you want to work for companies after graduation. Thank you. Now I would hand the PowerPoint to Steffi.
Yes, thank you to my team members here for presenting their very interesting biographies and their interests. And it's really exciting to see that each of you have different career plans and you enjoy different things in your courses. Very nice. So I would like to give you a little bit of background to my journey, how I ended up in biochemical engineering, and what I'm doing now. Right, so my journey started at the bottom left of this of this screen here, where after I've done my A-levels, I finished school, I wasn't 100% sure what I'm going to do. I really enjoyed biology. This is my pet subject at school. It was something I was finding really interesting, but I didn't want to study biology. I wanted to do something a bit more applied, so I decided to study nutritional sciences, and I did this in Germany because I was born and grew up in Germany. And so I went to the Friedrich Schiller University in Jena, started my degree, and after about a year and a half or so, I got to a point where I noticed that this isn't really my passion. I enjoyed some of the subjects, but really quickly found out that these were the subjects that were really science focused, like toxicology, biochemistry. I loved biochemistry, and I loved all the practicals that I did in my degree, but I didn't really enjoy so much the applied aspects of telling patients about nutrition and so on. I did work as a nutritionist for a little while to fund my degree. But I also did something, although I didn't enjoy the degree overall so much, I pursued some things that I really wanted to do. For instance, the courses I really wanted to do, I put a lot of effort in. And I also organized a 4-month study abroad trip to India with 2 other friends. And that was super interesting. And we worked in an agriculture university. And it had a really good time, very interesting time. But I realized pretty quickly that I want to do research and I want to do fundamental science, and to kind of come out of nutritional science and move into a slightly different area, I took up a research technician post. I applied to research technician posts in the UK, and I wanted to also learn English a little bit more than I had at school. And I got a job offered at UCL finally, and this is now a long time ago. So I took up that job just around the department I'm in now, and I worked on a very different area. I worked in wound healing, and we developed a wound healing product that is like a gel that is put on the skin, and that helped the wound heal fast. You can see a little picture here with skin samples. And my boss at the time also made a spin-out company from the work that we were producing. And I knew that I wanted to come out of this area and go into a little bit more technical and biochemistry focused fundamental research. And I wanted to do a PhD, and applied for PhD positions and got my PhD in biochemistry. 2007, I did work on making vitamin B12 in microbes, so in bacteria. And it was a really, really interesting project. And I had a little bit of synthetic biology focus too, but mainly biochemistry. So I was working on enzymes and how to put several enzymes together to make a very complex molecule. Vitamin B12 is one of the most complex molecules that exist on the planet. And then I took up a postdoc position in Kent University. In the meantime, I had a family. My oldest daughter was born, and then my twins were born. So really exciting times, and research and private life being really busy. And I then progressed into an academic career more and more, because I then also being a postdoctoral researcher, you do a lot of research in the lab, and it's a route towards an academic position. And I became a fellow. I was awarded a fellowship, and then from the fellowship, this was my kind of springboard into becoming a lecturer in synthetic biology at UCL, and this is now 3 years ago. So this is my journey, and really it's not been a straight line. It's been a little bit like this road here, you know, a little bit forth and back, and not all of this was planned, that's for sure. There were some opportunities that came along, and I was interested in these, and I grabbed them, and I went forward with these. But the job that was advertised at the time as a lecturer of SynBio was exactly down my path, and I knew this is gonna be for me, and I was really happy when I got offered. So what I'm doing at UCL, and a little bit what I've done before in my career, is my area in research interest is really in synthetic biology, biochemistry and synthetic biology, and my passion is looking into the molecular details of how you know, life works, and how the organisms work on the planet, and how we can actually make use of the properties they have. So in synthetic biology is not a very new field. It's been around for some time, but it's been accelerated over the last 10 years or so incredibly fast, because we can now do things with DNA that we will never be able to do 100 or 200 years ago. So we can take parts, let's say you can see bacteria here, or yeast cells here. We can take these microbes or plants or algae, and we can take properties that they have, and we can reprogram these organisms. For instance, in my PhD, I took bacteria, and I engineered them and programmed them, gave them a lot of enzymes that allowed them to make vitamin B12, which could be scaled up on a very large scale in industry to make it an industrial product, for instance, for older people to supplement their B12 levels and increase their uptake. But we can do many other things with re-engineering plants. For instance, we can get plants or algae to make biofuels for us, which all helps in connecting. Also, I should say, we're talking a lot in biochemical engineering about the circular carbon economy. For instance, we can also get plants to fix more carbon for us and make them available for food again, for example. But we can also, for instance, create artificial meat, or we can get microbes to make proteins for us that are used to feed animals. And this is a much more cost-efficient way of creating protein, rather than feeding cows on a large amount of plants and ground and soil and so on. Or and Rana and my other colleagues in the team have already talked about their efforts to engineer cells and stem cells, for instance, to treat cancer and other diseases, but also we can make other medicines, for instance, vaccines in bacteria and yeast, for example, or antibodies. And so these are biopharmaceuticals that we can make now on a quite large scale, and sometimes we even, well, we can also discover enzymes, for instance, in plants that do not exist in other organisms, and that we can take from the plant and put into an E. coli, for example, into a microbe, and we can get this microbe to make a product that only plants would be able to make normally. For instance, there's a very prominent example, which is making anti-malaria drugs in yeast. And normally these drugs are made by a plant, so yeast wouldn't normally in nature be able to do this, but in synthetic biology we can create new molecules in organisms that have never been able to do this. For instance, we can also make alternative solutions to plastics. Or in the case of our team this year, our team is looking into enzymes that come from several microbes to degrade plastic in the oceans, for example. Or we can make artificial fibers, materials, not artificial, but fibers that, for instance, would normally come from a spider, like spider silk, can now be made from yeast in a bioreactor, as Lee has explained a little bit before how bioreactors work and so on. So it's a huge potential in this area, and the global market of products coming from this in biofield is ever growing. Now, my personal research interest has been for some time now to take naturally existing organelles that exist in microbes, and you may not be so familiar with microbial organelles because they don't really get mentioned in a textbook, because traditionally they're not very well understood or known. But over the last 10 years or so, we have understood that microbes like bacteria also produce organelles, and these are called protein organelles because they're made entirely of proteins, as opposed to eukaryotic cells like mammalian cells or animal cells. They have lipid-bound organelles. But these protein organelles are very sophisticated. They can be made of many, many different proteins. They're quite big for a microbe like this, for instance, is a bacteria cell. And you can see these little blobs here. These are protein organelles. And I work with protein organelles from different microbes. You can see an E. coli here. You can see a Proteus mirabilis here, which can become a pathogenic organism in your genital tract. And I study these on a fundamental level. I understand how they work. This is my aim. But also, to understand how they work means you can then engineer them and make them do functions that they wouldn't normally do in nature. So, for instance, some years ago, we have engineered a protein microcompartment. You can see in purple, bottom left here. This would be one of these little blobs that you see on the top here. To actually make ethanol. So we created an ethanol bioreactor to make ethanol from feeding the cells glucose. You can make ethanol, so that could be used as a biofuel, for instance. And if you could grow this on a very big scale, you could make biofuels in E. coli. But there's many other applications. For instance, we can reprogram these particles for applications in vaccines. So we could make a vaccine based on these particles, or several types of vaccines, but also understanding how microbes use these particles. For instance, they use them for their own metabolism, for growing and outgrowing other microbes. For example, in our gut, we can use that knowledge to develop medicines or drugs for better gut health. And so there's many, many different applications and a lot of work in this field that we still got to do. But I'm also moving my research interest recently more into the vaccine area, where we're looking into, for instance, foot and mouth disease vaccines and other types of vaccines. And I thought a little bit about what kind of advice I can give you, and I've seen that our students have given you a lot of really, really nice advice on working, keeping an open mind. And my advice comes along a little bit along those lines too. What I have learned over the years is that, or advice I want to give you is be curious, explore the world around you, and work collaboratively, and try and be creative. So keep an open mind and see what else is out there. Even if you think is something in your degree that you don't like, or you've started something that's not quite down your road, what else is there to be taken from that degree? What else could you be doing? What do you really enjoy? Follow your gut feeling, your instincts, but also the things that you think would make you happy in the future, where you would like to work and which area. Really important, I found over the years, to me personally, is to work with many different areas and people in different areas, because together you're gonna be coming up with some really cool ideas that you may not think about yourself on your own. And a good example is our team, our iGEM team. We come from very different areas and programs and subjects, but we work together. We work modelers, work with synthetic biologists, work with people that like to work in the lab, and so on. And we come up with some unique ideas that we wouldn't have been able to think of ourselves, and come up with some new project. So yeah, and also put your creativity in. If you're an artist, see if you can combine your artist skills with your, with your scientific interests. I think a lot of good things come out of this. I personally like painting, so it's something I've always enjoyed doing alongside science. So this is my advice, and yeah.